Okay, so good morning everyone. I'm a master student, but I will not present my master project. It's because the Leishmaniasis is a very important disease, and we choose this topic because I think we must infer about this subject and this disease. So Leishmaniasis is an emerging and neglected disease. Leishmaniasis is an infectious disease caused by the flagellate protozoa, as Dr. Paulo Cotrin showed it before, like this, you can see here, genus Leishmania, and there is about more than 20 Leishmaniasis species related to human infection. The transmission is by the bite of the certain sandfly species, as we can see here. The sandfly is very small, it's 1/3 of the size of the regular mosquito, and it makes no sound when it's flying, and it's really hard to see, and it's really hard to feel the bite. So the transmission starts with the bite of the sandfly infected with the promastigotes, and the promastigotes will enters into the bloodstream, and the human body immune response will send some immune cells as a macrophage, and the macrophage will capture the promastigote, and the promastigote will fight back by hijacking the macrophage and change it to an oval form. This oval form is the amastigotes. The amastigotes will rapidly multiply inside the cell. It will blow up the cell and try to infect the other cells, and the tissues around the infection will start to die. And the cycle will continue with another sandfly biting this infected mammal, get the promastigotes, the parasite, and infected another mammal. The human is the mammal that we are concerned about, and the dog is the urban reservoir of this disease. So there are different types of Leishmaniasis: cutaneous Leishmaniasis and a systemic Leishmaniasis. But I put here 4 classical forms of the disease, the signs and symptoms of this disease. The cutaneous, you will see in the next slides, has a one sore of skin sore, and the diffuse cutaneous Leishmaniasis is a widespread skin lesions around the body, and the mucocutaneous Leishmaniasis is the sore that will commit the cartilage, mainly nose and mouth. And the visceral Leishmaniasis is the most severe form of the disease, potentially fatal. It's over the 90% fatal and lethal, and it will be on the inside organs, as spleen and liver. Here is a classical sore of Leishmaniasis. It starts after a couple of weeks, and it's a classical one. It starts around the mosquito bite, the sandfly bite, and it's like this. This is the diffuse form. It's pretty ugly to see, but it's like this. It's also it's a bad thing for the treatment because it's difficult to. And here the mucocutaneous Leishmaniasis, that we can see the cartilage damage. It's pretty ugly, and sometimes the patient cannot breathe, and it's also causes little death. And here the visceral Leishmaniasis without any sore, without any skin lesion, but we can see the enlargement of the spleen and liver. The spleen and liver. So the Leishmaniasis, it's confined in the tropical areas. And we can see on the in the green color the visceral Leishmaniasis, in the orange color the cutaneous Leishmaniasis on this map. And we can clearly see that's in tropical areas, subtropical areas, and southern Europe. There is 2 million people infected and 350 million people at risk. Here in Brazil, that is the situation. Visceral Leishmaniasis, it's almost in all states of Brazil, and cutaneous Leishmaniasis is already in all states of Brazil. So Leishmaniasis is a serious health problem, public health problem, because it's emerging. And it's an infection disease that incidence has been increasing in the past 35 years and could increase more in the future. it's also a neglected disease because it belongs to a tropical infection group of disease that it's endemic in low-income population and also in developing regions as Africa, Asia, and Americas. That it's no cases yet detected in USA, so you can be kind of tranquil. So the prevention measure, it's one of the measures that we are also concerned about because currently there is no vaccines used at a routine. And our national program of prevention and control of the disease, it's based on 3 steps. Control of the domestic reservoirs, the dogs, because those are the urban reservoirs. Controlling the vector and detection and treatment of human case. We do not treat the dogs. The procedure is the euthanize, it's sacrifice the dog. So that is the diagnosis. One of our challenge is the diagnosis because we have 3 types of diagnosis. Clinical diagnosis, the cutaneous it's kind of easier to see, but the visceral sometimes has a difficult because we can make sometimes it's like the same signals as another disease, and sometimes people just get confused. But the clinical diagnosis is about the signs and symptoms of the disease. Laboratory diagnosis could be serological, parasitological, and molecular tests, and epidemiological research, because we are looking for the areas of transmission. The laboratory diagnosis, it's based on the three classical types of diagnosis, parasitological, that will directly demonstrate the parasite on the cultivation or also in a stained slide. And serological tests, the most common ones are ELISA and IFA, that Dr. Paulo Cotrim mentioned before, and molecular diagnosis, the research of the parasite DNA. That's the mainly test used is the PCR, the polymerase chain reaction. Here we can see a stained slide with a mastigote forms inside the macrophage, the oval forms, and here we can see the pro-mastigote forms, the elongated form of Leishmaniasis. Here are ELISA, but I think you will see the ELISA test on our lab, and the immunofluorescence assay. It's almost like this. And the PCR, it's the amplification of parasite DNA, the polymerase chain reaction, and I can show you a short movie about this reaction.
The polymerase chain reaction, or PCR, amplifies a specific DNA fragment from a complex mixture. First, the mixture is heated to separate the DNA strands. 2 different specific oligonucleotide primers are added that are complementary to short sequence stretches on either side of the desired fragment. After lowering the temperature, the primers hybridize to the DNA, where they bind specifically to the ends of the desired target sequence. A heat-stable DNA polymerase and nucleotide triphosphates are added. The polymerase extends the primers and synthesizes new complementary DNA strands. At the end of this first cycle, 2 double-stranded DNA molecules are produced that contain the target sequence. This cycle of events is repeated. The mixture is again heated to melt double-stranded DNA. The primers are hybridized, and the DNA polymerase synthesizes new complementary strands. At the end of the second cycle, 4 double-stranded DNA molecules are produced that contain the target sequence. In the third cycle, the mixture is heated, the primers are hybridized, and DNA polymerase synthesizes new complementary strands. At the end of the third cycle, 8 double-stranded DNA molecules are produced that contain the target sequence. 2 of these molecules are precisely the length of the target sequence. In future cycles, this population increases exponentially. Cycle 4. Heating, hybridization, DNA synthesis. At the end of the 5th cycle, there are 22 double-stranded DNA fragments of the correct length, and 10 longer ones. Cycle 6, 10, 15, 20. After 30 cycles, there are over 1 billion fragments of the correct length, but only 60 longer ones. The product, therefore, consists of essentially pure target sequence.
So, as you can see, the diagnosis of PCR technique is more specific than the others, and it's not time-consuming, because the other one we need to wait for the promastigote multiplied and be isolated into the in vitro culture. So it's PCR, it's kind of faster than the other tests, and it's even more specific. And what's here? The treatment is kind of hard because there is a lot of side effects of the chemotherapies and the drugs that we are commonly used for the treatment of Leishmaniasis. And unfortunately, the parasite has become resistant to some of those drugs. And now, treatment is also a line of research that we are trying to develop it over and over. So our laboratory has mainly 3 lines of research. The parasite molecular biology, and we mainly use Leishmania. The molecular analysis of genes related with resistance and antiparasite compounds, as I show you, the parasite has become resistant to some compounds, some drugs, and new diagnostic approaches, because we need to improve the diagnosis to early identify the disease and treat it as fast as we can. So this is my master project, indeed. We are trying to evaluate the PCR technique as a molecular diagnosis with a specific identification of a serological agent of visceral leishmaniasis in different tissues of seropositive dogs. And it's also related with new diagnosis approach, because as I told you, the classical techniques for the diagnosis is also time-consuming, and it's hard to see the mSTGOT inside the macrophage with a stainless light. So our goal was to evaluate the efficiency of PCR in 5 different tissue samples collected from seropositive dogs and compare with the classical laboratory. That's why I can infer you that the PCR is the best one. And the city of Embudazats it's a tourist city, pretty close to São Paulo. It's only 23 kilometers away, 20 minutes to get there. And they have dogs infected with visceral leishmaniasis, the most severe form. The direct parasitological test and culture in vitro and clinical evaluation of the dogs were the tests that we compare. In Embudazat city, we study the profile of this tourist town in the Great São Paulo because it belongs to the Great São Paulo. We compare with the previous study to identify the sandfly species collected in the region. We also evaluate children between 4 and 10 years old, that those are the potential population at risk. We made a canine clinical analysis, and we also compare with those classical diagnoses as I showed. Here it's Embudazats, a map of Embudazats. All the colorful spots are infected dog. Was more than 2,000 dogs that we analyzed, and all those colorful spots they are confirmed with leishmaniasis, visceral leishmaniasis. But unfortunately, we could use only 26 of those dogs because it's not so easy to euthanize the dog and capture the dog. And here it's just a brief resume of my summarize of my research, that I can show you that the PCR was over than 80% efficiency and sensible than the other tests, and also faster than the others. So I would like to thank you all for the attention, and the entire team of our lab, especially Dr. Paulo Cotrini and Dr. Angelo Lindoso, I don't know if he's here, and the Center of Zoonosis Control of Embudas who help us with all the epidemiological research, FAPESP and SUSEM, the Soro Epidemiology Agency. So thank you very much.